All right, good morning everyone. Welcome to the very first session of OHBM. So my name is Alex Fornito. I'm from Monash University in Melbourne, Australia. And today, or this morning's educational workshop is going to be focused on trying to understand the brain from a network perspective, with a principal focus on using the tools of graph theory to map and model the brain as a connected network. And so I'll begin with a preliminary introduction to some of the basic ideas and how we go about building a graph model of the brain. And then the other speakers throughout the day are going to talk in more detail about specific aspects of how we go about analysing these methods, touching on different aspects of graph theory. So I guess as a starting point, you might ask, well, why would I want to think about the brain as a network, and why would I want to use a graph of all things to model the brain? And in case you've been living under a rock over the last 10 years, you've probably realised that neuroscientists have become increasingly concerned with mapping connectomes. And the term connectome was first defined by Olaf Sporns, Giulio Tononi and Rolf Kutter in this 2005 paper, and they defined it as a comprehensive structural description of the network of elements and connections forming the human brain. So the two key things in this definition are a structural description, so we're talking about anatomical connectivity, so effectively a wiring diagram of the brain, and a network description, right? So we're thinking about the brain as an interconnected network. And this makes a lot of sense because really, if you think about the brain as a multi-scale system with non-trivial levels of organisation at each scale of resolution, we can find networks at each of those scales. So at the molecular scale, we can map networks of molecules that drive synaptic signalling. At the micro scale, we can map networks of individual neurons and synapses. At the mesoscale, we can map axons connecting populations of neurons. And at the macro scales that we typically look at with human imaging, we can map large white matter bundles that link broader brain regions. And so then, given that the brain is organised across all of these different levels, it would be nice to have a single unifying way of representing these data, modelling these data and understanding these data. And this is what graph theory provides. And it can do so because it boils any connected system down to its essential elements. And so that's nodes and edges, right? And so the nodes represent the fundamental processing units of the network. So in the case of a brain, they could be neurons or populations of neurons. And the edges represent the interactions between the nodes. So in our case, some measure of structural or functional connectivity between the nodes. And so at a certain level, it might make you a bit uncomfortable to think we've got this extraordinarily complex organ such as the brain, and then we're going to use something as simple as a graph to represent it. But by focusing on just the core fundamental properties of the system, we can gain a lot in terms of the way we go about describing it and how we can think about it as just one example of a much broader class of complex systems. And this insight was first realised by Leonhard Euler, who is widely credited as being the founding father of graph theory. And he was a prolific mathematician, made a lot of foundational contributions in many different areas. But at the time, he was working on a problem called the Seven Bridges of Königsberg. And so Königsberg is now called Kaliningrad and is in modern-day Russia. And the town comprised a series of land masses interconnected by bridges, so shown here in red. And the problem was, is it possible to find a path to walk through each bridge once and only once without ever crossing a bridge twice? And Euler solved this problem by getting rid of all of the irrelevant details. So he ignored all the street locations, the topography of the land, and he represented the system as a graph. And so each landmass was represented as a node, and the bridges connecting them were edges. And so from this simple representation, he was able to distill the core features, and he deduced that no such path was possible. Then after his work, graph theory had some applications in some areas of chemistry mainly, and the next breakthrough really came in the middle of last century with the work of Paul Erdős, so another prolific mathematician. And he teamed up with another Hungarian, Alfred Renyi, to study the properties of probabilistic random graphs. And so these are graphs in which we just draw connections between pairs of nodes at random with some probability. And they were able to derive a number of analytic expressions that predicted various properties of the graph as you changed the probability with which nodes are connected. And so this then allowed people to start using this random graph as a model of many real-world networks found in nature, such as social networks, communication networks, and so on. But it really wasn't until the late 1990s where people started to think, well, maybe this random graph model isn't so appropriate for many networks that we find in nature, because they seem to show some properties that are distinctly non-random. And so one of the key insights was published in Science by Barabasi and Albert in 1999, and they focused on a property called the degree distribution. So the degree of a node is just the number of connections that are attached to it. And so if you plot the distribution of degree values across all nodes in the network, if you've got a random graph, you end up with a distribution that looks kind of like a Gaussian. Technically, it's called a binomial. And so most nodes have some modal or average degree, and the likelihood of finding nodes with very high or very low levels of degree drops off very rapidly, right? So most nodes are clustered around the mean. In contrast, what Barabasi and Albert noted was that many real-world networks have got a distribution that looks something like this. So most nodes have got very low degree, very few connections, but then we've got this extended tail here. And these nodes that are out in the tail, there's only a few of them that they've got a very large degree, much larger than you would predict from a random graph model. And so these networks act as hubs. They play a very influential role in the network because they're connected to a lot of nodes. And the network looks something like this. It's got this kind of hub and spoke organization. So specifically, Barabasi and Albert noted that this distribution follows a power law, and so they called these networks scale-free because there's a much deeper connection to power laws and a property of scale-free. And the basic idea is that there's no clear scale here. Unlike here, where the mean gives you a clear scale, here you just see this continuous progression from low degree to high degree, so they're considered to be scale-free. At almost the same time, Watson-Strogatz published another key observation in Nature, and they considered two different extremes for how a network can be organized. So on the one hand, we've got a completely random organization like that studied by Odosh and Renyi, and on the other, we've got a regular and predictable pattern. So here, each node is connected to its 4 nearest neighbors. And they asked the question, well, what happens if we start with this regular configuration and we just randomly rewire edges, and we do this one at a time until we get to this random configuration? And they studied two properties of the network. So one is called clustering. And clustering is essentially the fraction of closed triangles in the network. So you take any 3 triangles and you say, are they all connected to each other? Do they form a closed triangle? Or alternatively, they could form an open triangle like this. So you count the number of closed triangles and express that as a fraction of the total possible, and that gives you the clustering coefficient. And they developed this measure because I was thinking about social networks, and they thought, well, in a social network, if I'm friends with Bob and Tom, chances are Bob and Tom are going to be friends with each other because maybe we share the same interests, we go to the same party, and so on. So people tend to form local cliques of friendships. And this is not predicted by a random graph model. In a random graph, the probability that Bob and Tom are 2 friends should be the same as them being friends with anyone else. So they were interested in this property, and another property they looked at was the mean minimum path length. And that's the number of edges on the shortest path between 2 nodes. So if I want to go from this node to this node here, I go through 1, 2, 3, 4 connections to do so. So you estimate that shortest path for every pair of nodes, take the mean, and that's the characteristic minimum path length of the network. And so the mean path length is quite low in random networks. So you can see all of these shortcuts across the network. So if I want to get from one point to any other point in the network, I can do so in a short number of hops. Whereas if we look at the regular network here, you can see if I want to go from this point in the network to this point here, I have to go through many intermediary connections to do so. So the path length is very high in a regular network, low in a random network. But the regular network is very clustered, whereas the random network's not clustered. And what Watson-Strogatz found was as they randomly rewired the edges of the regular network, they found a regime in which you could just rewire a few edges, you get a dramatic drop in path length, but you preserve high clustering. So you get the best of both worlds. And they called this regime a small world regime, or a characteristic of small world networks. So the idea being that these networks are clustered like social networks, but you can find a path between any pair of nodes in a short number of hops. And this is an analogy to Stanley Milgram's famous experiment where he showed that people could find unknown strangers in social networks in just a short number of steps, giving rise to the famous 6 degrees of separation rule. So these small world networks, they've got high clustering and low average path length. And Watson Strogatz went on to show that many real world networks have got this small world combination of high clustering and low average path length, including the nervous system of C. elegans. So these two papers have had a profound impact, and they're among the most highly cited articles in science. They've got extraordinary citation rates because they really gave birth to this field of complex network science. And shortly after the other key finding was that many real-world networks show a modular organization. So this is the idea that nodes tend to cluster together into tightly connected subgroups or communities, for example, in a social network. And typically we see modules across lots of different levels of organization. So we find modules within modules at different hierarchical levels. A lot of other studies have gone on to show that real-world networks have got a lot of other non-random properties. And so this is why people have started to say, well, we can't just use the random graph model. We need to study real-world networks as they are. And so it's given rise to this discipline of complex network science. The idea being that a network is complex if it shows properties that are neither completely random nor completely regular. And if you think about it, the brain is probably the most complex network that we know of. And so then it makes sense that we could use the tools of this discipline of complex networks to try and understand what's going on in the brain. So if we want to generate a network model of the brain, and we want to do that in terms of a graph, then we need to define our nodes and our edges. And so to define nodes, in some cases, it can be pretty clear cut. So if we're looking at a neuronal level, we can say, okay, well, each node should be an individual neuron. But with brain imaging, we don't have that resolution, and so there's no real gold standard for defining our nodes. So we need to kind of just bear in mind a couple of criteria for what a good node in a network should be. And so one is that a node should be intrinsically consistent. So that means it should just represent a functionally homogeneous unit. So if I'm clustering a group of voxels into a node, those voxels should have some similar functional properties. The other is that nodes should be distinct from each other. So if I define a node here and a node here, they should have different functional roles in the network. And the third is in the brain, the node definitions should be spatially constrained. So we don't want a node to correspond to a region at the back and at the front of the brain. Our intuition about functional subunits of the brain is that they represent anatomically contiguous patches of tissue. So I guess the classic parcellation that tries to adhere to these criteria is Brodmann's cytoarchitectonic map. And this has been ported into the neuroimaging domain, largely by the work of Carl Zillies and Simon Eickhoff and team. So we've got probabilistic cytoarchitectonic maps, but only in certain regions of the brain. Alternatively, we could use variations in chemoarchitecture, so changes in receptor density and expression and so on. We could use anatomical criteria, so we could define regions based on sulcal and gyral landmarks. We could parcellate the brain at random using regions of different size. We could use functional criteria, so we could define regions of interest around peaks from, say, activation maps. Increasingly, people are getting interested in data-driven parcellation, so we try and cluster voxels together with similar connectivity profiles. We could use individual voxels as distinct nodes. We could generate myelin maps and use gradations in myloarchitecture. And last year, the Human Connectome Project published their version of a new parcellation of the brain based on an analysis of multimodal imaging data. So there's lots of different ways to define nodes. And my personal view is that there isn't really any single correct way. It very much depends on the hypotheses and the systems that you're interested in testing. But as a general rule, it is good practice to try and cross-validate your results against a couple of different parcellations, just to make sure that your findings aren't driven by the idiosyncratic properties of however you've decided to define your nodes. But assuming we're happy with our node definition, the next step is to define our edges. And so in the brain, we're talking about different measures of connectivity. And broadly, there's three classes of connectivity in the brain. So there's structural connectivity, which refers to the physical or anatomical connections between brain regions. This is intrinsically directed, so each axon has a source and a target, but commonly with imaging we can't resolve that directionality, so we can only tell where connections might be present in the brain. The next class is functional connectivity. So here we're thinking about brain activity, and functional connectivity is just some measure of a statistical dependence between physiological signals that we acquire in different brain regions. And so the key thing is this is estimated at the level of measured signals, and it could be directed or undirected. So I mean, the most famous example is a correlation coefficient, right? That's the most commonly used in the field. Then the final class is effective connectivity. And so this is the influence that one neuronal system exerts over another. And so the key thing here is that we're making inferences at the neuronal level. So if we've got an indirect measure of brain activity, such as bold fMRI, then we need some mapping from that measured signal to the neural dynamics that generated them. And so I guess a famous or common framework for doing this would be something like DCM, dynamic causal modeling. And so again, and in this case, we are looking at directed interactions. And so the main distinction between effective connectivity and directed functional connectivity is directed functional connectivity is estimated at the level of measured signals. Effective connectivity, we're trying to make inferences about neuronal population, neuronal interactions. Now, there's lots of different ways of measuring connectivity in different domains of neuroscience that vary in terms of how invasive they are. And people have tried to build connectomes and functional connectivity maps using all of these different techniques. I'm not going to go through all of them. I'm just going to focus on the two that we most commonly use in imaging, which is diffusion MRI for structural connectivity and functional MRI for functional connectivity. So we'll start with diffusion MRI. So most of you are probably aware that diffusion MRI measures the diffusion of water molecules in the brain. Axons provide barriers to that water diffusion, so water diffuses in the direction parallel to the trajectory of the axon. So we can use this information to propagate streamlines that try to track the direction that axons take in the brain. And there's different models of the diffusion signal and different ways of propagating these streamlines, broadly falling into deterministic and probabilistic tractography. And we can use these methods to generate whole brain maps of white matter fiber bundles. We then put our parcellation on top of that and work out which brain regions are connected to which. Now, there are a lot of different methods in this field with different strengths and limitations, which I'm not going to go through, but I will point out that in general there's a trade-off between specificity and sensitivity. So methods that are very sensitive in reconstructing tracks tend to be prone to false positives, whereas methods that protect you against false positives, they've got high specificity, tend to be prone to false negatives. And so we need to kind of determine where we want to sit on that trade-off. And Andrew Zaleski will talk a bit about how that trade-off impacts network analyses in the next talk. One thing I do want to point out is that with diffusion MRI, we typically estimate connection weights as either the number of streamlines that we reconstruct that connect two areas, or some measure of putative tract integrity averaged across that track, such as average fractional anisotropy. Now, these are very indirect measures of connectivity. So streamlines are not the same as axons, right? And our ability to reconstruct tracks depends on a lot of factors that aren't necessarily related to the physiological strength of a connection between regions. And for people who are interested in working in this field, I highly recommend reading this paper by Derek Jones that goes into the subtleties and complications of using diffusion MRI to measure anatomical connectivity. And it's important to keep these limitations in mind. I will point out that there is a lot of work being devoted to trying to develop more physiologically meaningful measures of anatomical connectivity using diffusion MRI. And these are going to, I imagine, proliferate throughout the community in the next 5 to 10 years. So now I'll talk about functional connectivity. So with functional connectivity, we record brain activity over time, so we get some measure of signal fluctuations over time in each of our regions or our network nodes. And so then we go and compute some measure of coupling between those signals. So it could be correlation, could be partial correlation, mutual information, whatever our preferred metric is. And so we do that for every pair of regions, and then we can build a network from those measures. Now, the critical thing with fMRI is to keep in mind the way we acquire the data influences the way we need to analyze it. So most commonly people do a standard resting state design, so we record these fluctuations over time, and then we summarize those dynamics with a single value, a single correlation coefficient. So it's kind of like a mean averaged behavior over that time period. And we're assuming that the brain is in a fairly stable psychological state and there isn't much dynamic behavior going on. More recently, there's been a lot of work trying to capture these dynamics in more detail. So looking at how networks evolve through time. So in this case, people use sliding window analyses, and you end up with like a time series of correlation coefficients. So then you can track how the network changes through time. Now, the critical thing in this work is that you really need to specify correct null models to allow you to determine whether the fluctuations you see are more than you would predict from simple random fluctuations, because random processes can fluctuate and look ordered in some way. So you need to have an appropriate null, and there's a bit of controversy in the field at the moment about the most appropriate null. But if you are interested in doing work in this area, it's important to familiarize yourself with that literature. And then the final domain is looking at task fMRI. And in task fMRI, it's not as simple as just taking the time courses and computing a correlation, because the task is driving brain activity in some way, and typically we're interested in trying to isolate the effect that the task is having on coupling, so the task-dependent coupling between regions, as distinct from kind of more background, intrinsic, task-unrelated processes, and also coactivation effects. So it's possible that stimuli could just drive activity into brain regions in a similar way without those regions communicating in any meaningful sense that would be implied by the term connectivity. So we need to use appropriate methods for trying to isolate that task-dependent connectivity, and there are methods for doing so. I won't go into those, but there are some references there for those who are interested. One thing that I do want to dwell on a bit is that the measures that we use to measure functional connectivity can introduce biases into the network analysis. So the standard Pearson correlation is the most common measure used in functional connectivity analyses. And this little schematic illustrates some of the biases. So let's say that I've got a network structure with anatomical connections that form a closed triangle between three nodes. Now in this case, a full correlation is going to do quite well and give you some estimate of the connectivity across those three edges. But then let's say that my structure looks like this. I've got an open triangle where B connects to A and C, but there's no connection between A and C. In this case, the correlation will give you some non-trivial estimate for the association between A and C, and that's just a mathematical property of the correlation coefficient, right? If A is correlated with B, B is correlated with C, then A and C need to be correlated as well. So the Pearson correlation tends to close open triangles, and as a result it leads to networks that are more clustered than they should be. So in this experiment we did a little while ago, led by Andrew Zaleski, we just took random numbers and correlated them, and we computed the clustering of those networks, and we compared them to networks with a random organization, and we found that these networks were more clustered than you would expect, and this is because of this what's called the transitivity of the correlation coefficient, this triangle closure property. And so that's introducing a bias into the network analysis, right? And so you might say, okay, well, all well and good, then I'll use partial correlations. And so in the case of an open triangle, the partial correlation will probably do quite well. It won't give you an estimate between A and C. But if the true anatomy is a closed triangle, then it's often hard to predict what the partial correlation will do. And it depends on the broader context. And we found that in large-scale network analyses, partial correlations lead to networks that are less clustered than you would expect by chance. So in some respects, they go too far and over-penalize and over-correct. So, there are measures that are kind of in between these, such as regularized inverse covariance techniques and so on. But it does mean that we need to think about the measures we're using to estimate connectivity and the biases we might introduce. And in some cases, you can use specific null models to try and account for these differences. And these are detailed in that paper cited at the bottom here by Andrew Zaleski. Right, but assuming that we're happy with our method for defining nodes and edges, then we want to build a graph model of the network. So we've got our nodes, we measure some aspect of structural or functional connectivity between every pair of nodes, And then we represent that in the form of a connectivity matrix, or what's called an adjacency matrix. And so here each row and each column corresponds to a different region, and each element corresponds to the type and strength of connectivity between pairs of regions. So for example, here element 1, 2 is the connection between region 1 and 2, 1, 3, between 1 and 3, and so on. And from this matrix we can build a graph representation of the network. And this is because matrices and graphs are mathematically equivalent. So just to kind of illustrate this point, here I've just got a simple binary undirected network, and here's the matrix representation. So you can see that node A is connected to E and C. And so if I go to this matrix, if I go to row A and column C, I find a connection. And I go to row A, column E, I find a connection. And that's in the upper triangle. If I go to the lower triangle, I can do the reverse. If I go to column A, row C, the connection's there. Column A, row E, the connection's there. This is because the network is undirected, so the upper triangle and the lower triangle are symmetric. They give us the same information. It tells us where the connections are. We're not trying to distinguish the direction that they're running in. If, on the other hand, we have a directed network, so we're looking at the direction of connectivity, the upper and lower triangles of the matrix are asymmetric. So in this case, you can see that there's a connection running from A to E, but not vice versa. And in the matrix here, we list outgoing connections down the columns. So if I go to column A, row E, I can see a connection. But if I go to row A, column E, there's no connection, right? So because we don't have that connection running backwards from E to A. So in this case, we're getting distinct information from the upper and lower triangle. And so this is just showing how the matrix and graph representations give you equivalent information. I should point out that that's just one convention of listing outgoing connections down the column. Some people do it across the rows. So if you're interpreting these matrices, you need to be aware of how the connections are represented. And then we can go on and represent connection weights. So in the graph, we can use differences in edge thickness, or we can use some color scale in the matrix, and we can do that equivalently for directed and undirected networks. So given these different graphs, we might think, well, what kind of graph best represents the brain? So on the one hand, we say, okay, well, it's a connected network, so we can use a simple binary representation, but we know that these connections vary in their strength, so we want to use a weighted representation. As I mentioned before, connectivity is intrinsically directed, so we want to account for directionality. And we also know that not all nodes are the same, so they vary in terms of cell density, gene expression profile, intrinsic dynamics, and so ideally we would want to represent that heterogeneity in some way as well. And we also know that connections are not the same. We have inhibitory connections, excitatory connections, modulatory connections, and so again, we'd want to think about representing this heterogeneity. So ideally, we're thinking about some kind of hybrid graph that combines all of these features. And we also know that networks evolve on timescales ranging from milliseconds to years. So we would want to be able to map all of these properties dynamically through time. Now, the nice thing about graph theory is it does provide us with a formalism and a framework for accounting for all of these features. But the problem is that our measurements are a bit limited. So most of the time, people have just focused on the simplest possible model, binary graphs. Increasingly, we're seeing people use weighted analyses, but particularly with MRI and imaging, it's a bit hard to accurately resolve directionality and edge and node heterogeneity. We're seeing a lot of connectomes in model organisms derived from invasive techniques that can do this properly. So we're going to be able to start to account for these features in more detail. And the nice thing is that graph theory provides us some room to grow. As our measures increase in sophistication, so can our models. But assuming we're happy with that, we've built our network, we've got our graph, so the next step is to then analyze the network. And we can do that in terms of either connectivity or topology. So connectivity is concerned with mapping variations in the strength and type of connectivity between regions, and so you could do, for example, a connectome-wide analysis where you're mapping effects at each edge in the connectome, and Andrew will talk about some methods for doing that in the next talk. Or we can look at topology. And so topological analyses are really concerned with trying to understand how connections are organized with respect to each other in the network. So really trying to unpack what are the key organizational principles of a brain network. And this is really where we use the mathematics of graph theory to gain insight into the network. And graph theory provides us with a lot of tools for doing that. And so the lectures for the remainder of this workshop are really going to focus on different aspects of analysis. So Andrew Zaleski will talk about how we go about thresholding networks and then the statistics that we can perform on networks. Bratislav Misic will talk a bit more about path length, diffusion models, and other measures of communication processes in networks. Sarah Muldoon is going to talk in more detail about modularity, how we go about understanding modules in networks from both a static and dynamic perspective. And Martin Vandenheuvel will talk about hubs, how we identify them, different ways of identifying which nodes might be particularly influential in brain network function. And then finally, Petra Vertes is going to talk about null models. So these are really important. They provide a baseline with which we use to make inferences on networks to determine whether a particular property is expressed to a degree greater than chance. And she'll also talk about generative models. So this is a very exciting and rapidly developing area where we try to explain a wide range of findings in a network from a few underlying simple wiring rules. So to summarize, hopefully I've convinced you that graphs provide useful models of brain networks that can be used at lots of different scales and with different types of data. There's different ways of defining nodes and edges, each with pros and cons, and these will constrain the way we interpret our findings. Brain networks can be either directed, undirected, weighted, unweighted, and there's also scope to incorporate additional elements in our models as we progress with our measures. And we can analyze networks at the level of connectivity or topology, and these analyses can be conducted at the level of individual edges, nodes, or at the level of the network as a whole. So it's quite a flexible repertoire of tools. So the slides of the talks, you should have gotten an email to be able to download them. And so here I've included some references. There are some general texts to graph theory and network science, as well as those more specific in the neuroscience context. Some software tools, in particular the Brain Connectivity Toolbox is a MATLAB toolbox developed by Mika Rubinov and Olaf Sporns that really gives you a lot of different functions for analyzing brain networks. Last year, Andrew Zaleski, Ed Bullmore, and I published a textbook on graph theory for neuroscientists, which is available through Amazon and Elsevier and might be available from the Elsevier booth here at the meeting. And also just a pitch for Network Neuroscience, which is a new journal with Olaf Sporns as editor-in-chief that's accepting submissions for any network analyses in neuroscience ranging at all scales from molecular to macro. So if you're active in the field, please consider sending your work our way. But thanks for your attention, and I think we've got time for one or two questions. So please come up to the microphones if you've got any questions.
Thanks for the talk. It's really great. I was just wondering, you mentioned the limitations of the correlation, partial correlation, and the triangles, how the correlations might not be the best description of the structural connectivity, and I was wondering if you've seen any work of like doing some kind of inverse weighting with like path length between structural, like with structural data, to kind of multiply the matrices and get like a better idea and address those limitations of correlations between functional areas.
So using the network structure to constrain the functional connectivity estimates?
Right, so like just getting like a structural matrix, using something like just a binary or inverse path length or something between areas using structural data, multiplying it by the functional, and then kind of getting over the limitations of like the correlation and partial correlation.
Off the top of my head, I can't think of anything that's been done in that domain.
Okay.
Yeah, I guess you'd probably have to think about exactly how you'd do the mapping and how you'd incorporate it as a prior in the model. Yeah.
Okay, cool. Thank you.
Thanks for the great overview. So you said the link will be available somewhere. That's my first question.
Yes, I think people that registered for the educational workshops would have been emailed a link to be able to download the talks. Yeah.
And my second question is about this, again, for the Pearson and partial correlation matrix. So if you think about directed connectivity, which we generally use in EEG or MEG, for example, let's say coherence, and if you have such a directed network always, then always we end into problems that we cannot have all the direction for the whole brain. So that's why I think graph is a really nice measure to get it done. So do you think we can use those as a, instead of the correlation matrix to get the connectivity?
I mean, I think it boils down to how much you believe your functional connectivity measure. And I think with directed functional connectivity, it is a bit trickier. So the further removed you are from the neuronal signal, the harder it is to, I guess, believe the directions. So if you're looking at LFPs or spike counts and you're doing, I don't know, Granger causality, perhaps, you know, that's appropriate. But when you then go out to EEG, MEG, fMRI, it gets a bit trickier. And I guess there's a bit of controversy in the field as to the best way of doing that. And I think that's probably the biggest limitation. There's a lot of information we can get by having directed connectivity that we miss a lot of the time. So.
And the last question is, what do you think about the multimodal? How can you integrate different modalities? Which would be the best option?
I think it depends on your question. So it depends on the hypothesis you want to test. So, you know, Sarah later today will talk about multilayer networks, which kind of allow you to have a unified way of analysing different types of networks. So that could provide one framework. But, you know, I guess it depends on how specific you want to be in linking structure to function. I don't think there's a one-size-fits-all approach.